Hello, my name is Aissa Houdjedj, and great to have a pleasure to be here today. First of all, I want to thank the organizers for giving me the opportunity to present my talk, which is titled Ranking Cancer Drivers via Betweenness-based Outlier Detection and Random Walks. Now, so the cancer is caused and developed by a subset of somatic mutations that are called drivers. So our main challenge here is to construct a new method that integrates genomic and connectivity information to identify the small number of driver genes from the background that consists of large set of passenger mutations, in which later on it could help in the development of treatment and therapies. And our constructed method is compared against 5 other methods. The first method is Betweenness, in which our method is inspired by. So they employ paired data and interaction network, and they use Betweenness measurement to prioritize cancer genes. And our main contribution is to use Betweenness measurement for the identification of patient-specific dysregulated genes on personalized networks. DriverNet, which is a computational framework that prioritizes mutated genes based on their degree of network connectivity to a set of dysregulated genes in the tumor samples. Downrank and subsequency are the methods that extends the idea of DriverNet, and they perform a direct random walk in the prioritization framework. And finally, IntDriver is using interaction network and gene ontology data within a matrix factorization framework. And now here I will be describing our method. As an input for each sample, we have gene expression measurement for both normal and tumor tissue. Also, a binary mutation matrix is constructed in which we exclude the silent mutations. And to construct the pan-cancer data, we take the union of all the samples from cancer types that have more than 10 paired measurements from both normal and tumor samples. And in addition to that, we also perform separate evaluations on breast cancer and lung cancer. We also employ protein-protein interaction data from the IntAct database that consists of 15K nodes and 113K edges. Now, the first step of our method is the Bituminous calculation. So for each sample, we will have 2 graphs, normal and tumor graph, that consist of the PPI network at the beginning. Then for the normal graph, we remove non-expressed genes. And for the tumor graph, we also remove non-expressed genes and the mutated genes as well. After that, we run Bituminous calculations on both normal and tumor networks. So the idea of Bituminous centrality is measuring the extent to which a vertex is lying on paths between other vertices. And its standard definition is ignoring the length of shortest path. And by considering that very long paths as functional relations might not be biologically meaningful, we are using a k-Bituminous versions on which we consider only paths of length less or equal to k. Then after the Bituminous calculation is done, for each gene I, you calculate the absolute difference of Bituminous centrality values calculated from the normal and tumor networks. Next step is the selection of outlier. So for each gene I, we plot the Bituminous values across all the patients, and we estimate the standard deviation and the mean by fitting a truncated normal distribution. And a gene I is selected as an outlier in patient P if its Bituminous difference value greater than T standard deviations from the mean. To finalize the Betweenness step, a bipartite graph is constructed that consists of mutations in one partitions and a set of outliers in the other partitions. And an edge is drawn between an outlier and the mutated gene if there exists such an edge in the PPI network. Next step, a random walk is applied on the bipartite graph with restart probability, and the mutation frequencies of the genes are assigned as initial hit values to be diffused during the random walk. Then, our final step is ranking genes, and to rank mutated genes of the bipartite graph, we are using the following formula. The score of mutated gene is equal to the alpha multiplied by the degree of the mutated gene I plus 1 minus alpha multiplied by the weight of the incoming edge of the mutated gene I. And the weight of the incoming edges of the mutated gene I is equal to the sum of the incoming edge weights for gene I after the random walk. And gene I with the largest score is chosen as the next driver. Then we remove the corresponding node and its neighbors from the bipartite graph and recompute the scores again until we cover all the outliers. So to evaluate our results, we compile non-cancer genes from CGC and CancerMine databases. And CancerMine is a literature-minded database of cancer genes in which we decided to include it as it is a cancer type specific reference set. And we also identify the CGC genes with mutation frequencies less than or equal to 2%. We call them rare drivers. Then we started evaluating our methods with respect to reference cancer gene sets by computing true positive and false positive rates for the top 1000 ranked genes. Then, according to the ROC curves obtained from lung cancer dataset, we observed that our method, which is named Betweenness, is performing better over all evaluations here. And we also observed the same thing from breast cancer dataset, in which our method is performing better over all reference sets that we have here. Lastly, for pan-cancer dataset, we observed that our method ranks the second for both CGC and cancer minor reference set. And for CGC rare, our method and downrank are having similar performance, and they perform the best. We also tried another evaluation, in which we define new metrics that measure how well the associated functions of the ranked genes and the reference set match. So first of all, we find the enriched Go terms in the ranked gene set who vary in total sizes from 100 to 500 in an increment of 100 for each method under consideration. And also, we repeat the same Go term enrichment analysis with reference gene set. And we calculate the Go or a consistency score, which is the size of intersections of Go terms divided by the size of the union of Go terms. For the Go evaluation, we observed that our method ranked genes is performing the best for lung cancer for almost all total size values. And for breast cancer, downrank ranks the best in 4 out 5 total size values. And our method is the second best. And for pan cancer, as you can see here, it's really, there is no clear winner here because driver net, Betweenness, and downrank are performing close to each other. After that, we repeat the same type of analysis by replacing Go enrichment with pathway enrichment. In this figure, the results are with respect to reactant pathways. For lung cancer, as you can see, our method outperforms the other methods with a large margin for total size 100 and 500. And for breast cancer, our method performed the best for almost all the total size values. And for pan cancer, our method is given the top consistency value in 4 out 5 cases. As a conclusion, our main contributions is the identification of patient-specific dysregulated genes with the major based on the Bituminous centrality on the personalized network, and also the employment of random walk on the resulting influence Bipartite Graph. And we have shown in our publication that many of our top-ranked genes, that they do not appear in reference cancer genes, they have roles in the cancer development based on the existing literature. Last, I would like to thank all the members of Antalya Computational Biology Lab, especially my advisors, Jesse Martin and Hilal Kazan, and also our sponsors for their contributions. Thank you so much. If you have any questions, thank you.